Good day, students. In this video, I will discuss about proteins. You see, nearly every dynamic function of a living being depends on proteins. A human has tens of thousands of different proteins, each with a specific structure and function. Proteins, in fact, are the most structurally sophisticated molecules known. But what really are proteins, and what are proteins made of? Proteins are the most abundant biological macromolecules present in all cells. As a matter of fact, proteins account for about 30 to 50% or even more of the dry mass of most cells. Proteins are versatile organic molecules composed of polymers of amino acids that are covalently linked by peptide bonds. The next two slides will show the important functions of proteins. First, enzymatic proteins such as digestive enzymes accelerate certain chemical reactions. Digestive enzymes catalyze the hydrolysis of bonds in food molecules. Proteins also play defensive roles in the body. For example, antibodies have the capability of inactivating bacteria and viruses, consequently protecting the body against diseases. Storage proteins help in the storage of amino acids, and transport proteins such as hemoglobin functions in the transport of oxygen in the body, and there are also some proteins that help transport molecules across the cell membranes. In addition to this, there are also proteins that help in the coordination of an organism's activities. Hormonal proteins such as insulin help in blood glucose regulation. And also, receptor proteins have important roles in cell signaling cascades. Lastly, contractile and structural proteins provide movement and give support to organisms, respectively. Now let me talk about polypeptides. So diverse as proteins are, polypeptides are all unbranched polymers constructed from the same set of 20 amino acids. Polymers of amino acids are called polypeptides, and a protein is a biologically functional molecule that consists of one or more polypeptides, each folded and coiled into a specific three-dimensional structure. Now let me talk about the amino acid monomers and its structure. All amino acids share a common structure. An amino acid is an organic molecule possessing both an amino group and a carboxyl group. The illustration at the bottom of the slide shows the general formula for an amino acid. At the center of the amino acid is an asymmetric carbon atom called the alpha carbon. Its four different partners are an amino group, a carboxyl group, a hydrogen atom, and a variable group symbolized by letter R. The R group, also called the side chain, differs with each amino acid. The physical and chemical properties of the side chain determine the unique characteristics of a particular amino acid, thus affecting its structural role in a polypeptide. So amino acids are grouped according to their side chain properties, and I will be giving the examples of each classification or each group of amino acid in the next two slides. One group consists of amino acids with non-polar side chains, which are hydrophobic. These amino acids with a hydrophobic side chain include glycine, alanine, valine, leucine, isoleucine, methionine, phenylalanine, tryptophan, and proline. Notice that each amino acid is assigned a 3-letter and a 1-letter acronym. Another group consists of amino acids with polar side chains, which are hydrophilic. These are serine, threonine, cysteine, tyrosine, asparagine, and glutamine. Acidic amino acids are those with side chains that are generally negative in charge owing to the presence of a carboxyl group which is usually dissociated or ionized at cellular pH. Basic amino acids have amino groups in their side chains that are generally positive in charge. Because they are charged, acidic and basic side chains are also hydrophilic. Now that we have examined amino acids, let's see how they are linked to form polymers. When two amino acids are positioned so that the carboxyl group of one is adjacent to the amino acid of the other, they can become joined by a dehydration reaction with the removal of a water molecule. The resulting covalent bond is called a peptide bond. Repeated over and over, this process yields a polypeptide, a polymer of many amino acids linked by peptide bonds. The pioneer in determining the amino acid sequence of proteins was Frederick Sanger, who, with his colleagues at Cambridge University in England, worked on the hormone insulin in the late 1940s. Because of his work in determining the sequence of amino acids that form insulin, this allowed other researches on determining the structure and function of proteins to be conducted. So a protein's specific structure determines how it works. Currently, there are 4 levels of protein structure known. These are primary, secondary, tertiary, and quaternary structures. The primary structure of a protein is a linked series of amino acids with a unique sequence. As an example, let's consider transthyretin, which is a globular blood protein that transports vitamin A and one of the thyroid hormones throughout the body. Transthyretin is made up of 4 identical polypeptide chains, each composed of 127 amino acids. Shown here is one of these chains unraveled for a closer look at its primary structure. Each of the 127 positions along the chain is occupied by one of the 20 amino acids indicated here by its 3-letter abbreviation. The primary structure is like the order of letters in a very long word. However, the precise primary structure of a protein is determined not by the random linking of amino acids, but by inherited genetic information. The primary structure in turn dictates secondary and tertiary structure due to the chemical nature of the backbone and the side chains of the amino acids positioned along the chain. Most proteins have segments of their polypeptide chains repeatedly coiled or folded in patterns that contribute to the protein's overall shape. These coils and folds, collectively referred to as secondary structure, are the result of hydrogen bonds between the repeating constituents of the polypeptide backbone. Individually, these hydrogen bonds are weak, but because they are repeated many times over a relatively long region of the polypeptide chain, they can support a particular shape for that part of the protein. While secondary structure involves interactions between backbone constituents, tertiary structure is the overall shape of a polypeptide resulting from interactions between the side chains or the R groups of the various amino acids. The tertiary structure of the protein is contributed and reinforced by hydrophobic interaction and disulfide bridges. Some proteins consist of 2 or more polypeptide chains that are aggregated into one functional macromolecule. The quaternary structure is the overall protein structure that results from the aggregation of these polypeptide subunits. For example, shown here is the complete globular transthyretin protein, which is made up of its 4 polypeptides. Another example is collagen, which is a fibrous protein that has 3 identical helical polypeptides intertwined into a larger triple helix, giving the long fibers great strength. This suits collagen fibers to their function as the girders of connective tissue in the skin, bone, tendons, ligaments, and other body parts. Hemoglobin, which is the oxygen-binding protein of red blood cells shown in this slide, is another example of a globular protein with quaternary structure. It consists of 4 polypeptide subunits, which is composed of 2 of the alpha subunit and 2 of the beta subunit. Both the alpha and beta subunits consist primarily of alpha helical secondary structure. Each subunit has a non-polypeptide component, which is called heme, with an iron atom that binds oxygen. Even a slight change in the primary structure can affect the protein's shape and ability to function. For instance, sickle cell disease, which is an inherited blood disorder, is caused by the substitution of valine for the normal amino acid, which is glutamic acid, at a particular position in the primary structure of hemoglobin. Normal red blood cells are disc-shaped, but in sickle cell disease, the abnormal hemoglobin molecules tend to crystallize, deforming some of the cells into a sickle shape. This is the end of my discussion on the functions, composition, and structure of proteins. Thank you for listening and have a great day, everyone.